Hello everyone, welcome back. I hope I'm clearly audible and visible to all of you. So, up till now, we saw that how Mendel, all right, we studied in the previous chapter about Mendel basically, and we saw how he set the foundation for genetics with his experiments of pea plant, and we saw that how he said that it was the genes that or genes or the chromosomes that were transferred from one generation to the another. So there were traits transferred from one generation to another. All right, now uh it's just a moment. Fine. So now in this chapter, this is the unit 6 for sorry, this is under the topic unit genetics. The chapter name for the NCRT is molecular basis of inheritance. All right, fine. So let us start. I'm Janani Saptharishi and I'm associated with Unacademy. Unacademy is India's largest online learning platform. Hi Ayushi, how are you? So all your doubts you will be posting it in the chat section. All right? So, what Unacademy will give you? Unacademy will give you daily live classes. All right. So, what happens in here that every day you will get access to all the daily live class. All right. So, So now you'll avail every day, you will be able to avail all the daily live classes. You'll have live tests, live quiz in which you can evaluate your performance. So what happens is in the daily live classes, all your doubts, you can engage in all the doubt clearing sessions and what all is going on. It is a very interactive session. Then comes that we have a very structured course. So we have planned the course according to the exam you wish to crack. So Unacademy offers you the preparation for any entrance exam that is offering across India. So all our structured courses are in line with your exam syllabus, and you can prepare uh, with the best educators that are on board. We have unlimited access to all the contents, and we have live and recorded lectures. You can watch at your comfort, at your device, at complete ease. All right. So what happens is, see, if you are taking and appearing for an examination like NEET, so it is not that if you will use my code, you will avail only contents of biology. No. See, Unacademy offers you complete syllabus coverage. By that I mean you'll get access to physics, chemistry, biology, and all the subjects that are required to crack NEET. Now, we have daily live classes, and you will be on board with the top educators of India. So they will help you to get in track, and they introduce you to a very comprehensive syllabus. All you need to do is go to the Unacademy Learning app and get the subscription. Once you do that, this is the fee structure that is offered to you. I recommend you the most the 12-month plan. Why? Because it is very economical. So you get to avail 12 months of learning in just 15,000 rupees. All right. And uh, apart from that, you uh, Hi Arun. All right. So if you're in your class 11th, you should highly go with the 12-month plan. So to crack an examination like NEET, you at least need 12 months in hand. And if you're in your class 12, you can go for the 6 or the 3-month plan that offers you the crash courses. Now, use my name, that is the code Janani, and from that, use this as your referral code, and you will get an extra 10% discount on all these fee structures. So you can avail an extra 10% discount by using the referral code Janani. All right. The search for the genetic material. So this will be our topic for today. So as I said, now from we had discussed in chapter 5 about Mendel. This is the chapter 6 of the NCRT under the unit 6 genetics. So the search for the genetic material began after. Hi Indu, hi Uruj, hi Harman. So, the search for the genetic material began after Mendel's said about the traits and all the factors that are moving from one generation to the another. All right. So, now they saw that what it was the thing that is flowing from one generation to the another. What was the genetic material? So Gregor, Mendel, Watson, Sutton, Thomas Hunt Morgan, and the other scientists narrowed down their search to the chromosome. All right, that is, पहले मेंडल ने क्या कहा था कि he did not specify chromosome. He only stated that there is a transfer of some material from one generation to another. So Walter and Sutton came up with the chromosomal theory of inheritance. All right. Then came Thomas Hunt Morgan or T.H. Morgan, who verified Walter and Sutton's theory. All right. And other scientists narrowed down their search to the chromosomes. So, as he said, Walter and Sutton, in the chromosomal theory, they said that the chromosome is what contains the gene. All right. And they are the one that are transferred from one generation to another. So, the chromosome located in the nucleus is in the, where is this chromosome located? It is located in the nucleus. All right, fine. So, coming to the transforming principle, or how this person, Griffith, this transforming principle is also known as the Griffith experiment. So, this scientist is very known. From this experiment, we got a hint of the genetic material, that is the DNA. So, he basically stated a transforming principle in which he said that there was some material that got transformed and it flowed from one generation to the another. So the transformation experiment was first performed by the British bacteriologist Frederick Griffith in 1928. So he carried a series of experiments with bacteria. All right. So what did he use for his experiments? He used a bacteria called as Streptococcus pneumonia or pneumonococcus. All right. Fine. So, and this is also known as Diplococcus pneumonia. But for your examinations, please remember this name. It is very important, Streptococcus pneumonia. All right. So this parasite is basically pathogenic. All right. By pathogenic, what I mean that they cause the disease called as pneumonia in mammals, including humans. So S pneumonia, as the name suggests, will cause pneumonia. And what is it? It is a bacteria. Now, what did Griffith do? That this Streptococcus pneumonia has two strains. All right. One is the S strain and one is the R strain. So he studied both of these strains. So the S strain is basically, you have to remember that it is virulent or pathogenic in nature. That means it will cause the disease. And it will basically, it has a covering or a mucilage. All right. So mucilage is basically a thick covering outside, which is smooth and regular. So S refers basically to smooth and R refers to rough. Clear? So S refers to the smooth or the regular colony, and it has the ability to cause disease of pneumonia. And the R strain is non-virulent. By non-virulent, I mean that it is non-pathogenic. That means It does not have the ability to cause the disease. It is unable to cause the disease, and it is exactly opposite of that of the eggs. So it will lack mucilage, it will lack the rough and the irregular colony, and it will not cause the disease. So S stands for the smooth covering, that is all right, and the R stands for the rough covering. All right. If you have any doubts, you can post it in the chat box. Fine. Series of experiments of Griffith are as follows. So what did Griffith do? He basically injected the S strain into the mice. So he took the mice and injected the S strain, and the mice developed the disease pneumonia. Why? Because we saw that S is pathogenic and S is able to cause the disease. And eventually, as it is pathogenic, it led to the death of the mice. So the living S strain contains the living S cells are recovered. That is, the body will contain the S cells of the, so the dead mice body will contain the S cell. All right. So now एस सेल को जब हमने इंजेक्ट किया तो माइस मर गया. ठीक है. अभी हमने आर स्ट्रेन को इंजेक्ट किया माइस के अंदर, and the mice did not get the disease. Why? Because R is non-pathogenic, and the living R cells was recovered from the surviving body of the mice. All right. So when S strain was injected, the mice died, and when R strain was injected, the mice survived. R is non-pathogenic. Yes, Sindhu, you are right. So, Griffith was able to kill the bacteria by heating them. तो अभी ग्रिफिथ ने क्या किया कि heat killed S strain लिया, यानी कि he took the S strain and just heated it. So by heating, what happened? The bacteria got killed, and the bacteria, when ejected into the mice was not killed. That is, the mice survived. So when he ejected a mixture of heat killed S and live R, here comes the interesting part. All right. तो अब तक हमने देखा कि एस सेल्स को जब इजेक्ट किया माइस में, सो द माइस सर्वाइव्ड. ऑल राइट. नाउ आर सेल्स में क्या हुआ? माइस डाइड. दिस इज सर्वाइव्ड. नाउ व्हाट हैपेंड? हीट किल्ड एस सेल्स. Mice was able to survive. Why? Because the S cells were killed by the action of heat. But when the R cells and the heat killed S, heat killed S, all right, was ejected, the mice died. So what do we conclude here? See, what happens here? R cells say mice is, sorry, R cells say, I wrote it wrong. I'm so sorry. With the S cells, the mice died, and with the R cells, the mice survived. All right. So R cells से mice survive हो रहा है. All right. Heat killed S से mice survive हो रहा है. तो जब भी R cell and heat killed S cell दोनों साथ में डालेंगे, तो basically mice should survive, because R cell is also is non-pathogenic and heat killed is also non-pathogenic. All right. So by that, it should basically survive. But what we see in the result is that when we combine both of them, it is the mice dies. All right. So what happened when the heat killed S and the live R is ejected together? All right. Both are non-pathogenic, but isse mice is dying. All right. So this is an unusual thing that he observed. And what did he recover? From the mice, dead mice, he recovered the living S bacteria. All right. So what did he see when the mice died because of this? Living S bacteria जो है, that was recovered from the mice. So now what happened? He injected heat-killed S strains into the mice, and mice did not get the disease and remained alive. Alright? So, heat-killed S से survive हुआ, but जब दोनों साथ में आया तो pneumonia हुआ and it died. So, Griffith, basically what he concluded was that the R strain had somewhat transformed itself. Alright, so what did he use the word? He used the word transformed. That the R strain somehow transformed himself into the S strain, alright, by the heat killed S, alright. So that he concluded the transforming principle, that is the transformation, or how the how somehow the transforming principle, that is transfer from the heat killed S strain, had enabled the R strain to synthesize smooth polysaccharide coat. That means the R strain is basically transforming into the S strain. Alright. So this is what Griffith observed, and he named this as the transforming principle. So this he told that it can be due to a genetic material, but it was not known with this experiment that the genetic material was DNA. So later, after 16 years, after 16 years, there came three scientists, that is Avery, MacLeod and McCarthy, who told us that it is the DNA that is undergoing the transforming principle, or DNA is the one that is transferred from one generation to another. So what happens here? However, the biochemical nature of the genetic material was not defined from his experiments. Alright, that means कुछ ऐसे एक मटेरियल हम कंक्लूड नहीं कर पाए कि क्या जेनेटिक मटेरियल है और क्या ट्रांसफॉर्म एंड ट्रांसफर हो रहा है. So what happened was, prior to the work of Oswald, Colin, McLeody, and MacLean, so the genetic material was thought to be a protein. Alright, so now came three scientists, Oswald Avery, Colin MacLeod, and MacLean McClarty. What they did, so these scientists, after 16 years, they worked on the same, they worked, or I should say continued, Griffith's experiment to determine that what was the biochemical nature, or what was the substrate, what was the thing that was getting transformed. So basically, they worked on the biochemical nature of the transforming principles. So what they did, they purified. Hi, Ravi sir. So, what they did was they basically purified the biochemical proteins, DNA, and RNA from the heat killed S cells to see which one could cause the transformation. Alright. Now, what happened? They discovered that the DNA alone from the S bacteria caused the R bacteria to be transformed. So, what happened was they also discovered that the protein digesting enzyme, proteases, RNA digesting enzyme, that is the RNAs, did not affect the transformation. So, what they basically did, let me show you. So, what they did was they took a test tube. All right, in this was basically they had the S cells that were heat killed. All right, so they had S cells in a form of solution, or the S strain, I should say, in a heat killed form. All right, so what they did was they removed. So from the solution, lipids and proteins were removed out. All right. तो लिपिड्स एंड प्रोटीन्स को इस सॉल्यूशन से निकाल दिया. Then what did we observe? Then we saw that three enzymes. So they basically worked with enzymes. So three enzymes were added to it. What were the three enzymes? Let us see. So the three enzymes that were added is proteinase, RNase, and the DNase. Alright. So these were the three enzymes that were added. So then what happened was, तो अब क्या हुआ कि जब प्रोटीनेज को इंट्रोड्यूस किया तो बेसिकली व्हाट हैपेंड वाज we could not see any, in case of proteinase, what it does, it will kill all the protein, so we cannot find any protein here. तो क्या हुआ, but still in the solution, all right, so जो solution है हमारे पास, we could see the presence of the S strain. All right. So basically, if proteinase was the transforming enzyme, what would have happened? So if protein was the transforming enzyme, तो प्रोटीनेज से ये प्रोटीन किल हो जाता एंड यहाँ पे सिर्फ R cells present है, ठीक है. And when proteinase was introduced, we could, हमें ये नहीं, R cell से S strain का transformation नहीं दिखता. All right. So now coming to the RNase. RNase में क्या हुआ कि it killed all the RNA. It killed the RNA. Here what happened, that we have this, these are the R cells. All right. So now what happens in the case of RNase also, similarly, the S strain appears. All right, strain S will appear. The S strain will look something like this. So this appears. So RNA is also not the transforming enzyme. But in the case of DNA, what we observe is the DNA is basically what happens, that when DNA is acted, all the DNA is now killed. Or instead of killed, I should say that all DNAs basically will remove all the DNA. All right. So basically DNAs has removed all the DNA. Now what happens? Here we cannot see any S strain. No S strain is visible. What you will see? You will see only the R cells here. So the R cells have not transformed themselves to the S strain. This is clear? So here hence we conclude that it is the DNA that will undergo the transformation. Alright. So basically it is the DNA that will help in the transformation. So in the absence of the DNA, there was no transformation from the R strain to the S strain. This is clear? अब proteinase के case में protein was killed or protein was removed out, but there was RNA and there was DNA. So transformation took place and we could see the S strain. And similarly, in the case of RNAs, the RNA was removed, but there was DNA and there was protein. So DNA again helps there to transform them from the R to the S. But in the case of DNA, the main genetic material that is required for transformation is not there. All right. Now comes the next experiment of Hershey and Chase. Here what happens, that the unequivocal proof that a DNA is the genetic material came from the experiment of Alfred Hershey and Martha Chase. Now what they did, they worked with the virus that infected the bacteria. So they basically worked with a bacteriophage. So what they did was, the bacteriophage is basically attached to the bacterium. All right. And the genetic material will enter from the bacterial cell. The bacterial cell, when it is treated with the viral genetic material as if it was by its own and subsequently manufactures more virus particles. So what happens, the bacterial cells के अंदर ये viral genetic material अपने आप को replicate यानी कि manufacture करना start कर देता है. And they try to observe which component of the bacteriophage, that is the either the protein or the DNA, entered the bacterial cells. So again they are testing कि क्या enter हुआ bacterial cells में. Was it the protein that entered it or was it the bacterial cells? And they directed the reproduction of the viruses. Alright. So what happened here? Hershey and Chase grew the virus on a medium that contained radioactive phosphorus. So the radioactive phosphorus, they took 32P, and some other on the medium that contained radioactive sulfur, that is 35S. So in दो radioactive material पे virus को grow किया गया था. Alright. Then what happened? When the virus was grown in the presence of the radioactive phosphorus, what did we see? They contained radioactive DNA, because DNA contains phosphorus but the protein does not. तो अब हमने क्या देखा कि वायरस को जब भी ग्रो किया गया था इन द प्रेजेंस ऑफ रेडियोएक्टिव फॉस्फोरस. ठीक है, फॉस्फोरस क्यों लिया हमने? एक्चुअली हमने दो एलिमेंट लिया, फॉस्फोरस एंड सल्फर, और डीएनए में फॉस्फोरस इज प्रेजेंट एंड सल्फर इज not present. So if the radioactive, if we know that the DNA is present, then the virus will be able to grow in the medium of phosphorus. All right. So now what happens? हमें वायरस के अंदर से क्या मिला? फॉस्फोरस मिला, यानी कि we concluded it because DNA is what contains phosphorus. All right. Now, the radioactive phages were allowed to attach to the E. coli bacteria. What happened then? Then, as the infection proceeded, the viral coat was removed from the bacteria and they were now agitated in a blender, or they were centrifuged. Then what happened? The virus were separated from the bacteria by spinning them in a centrifuge. What is a centrifuge? Basically, we spin it, all right, in a very high rotation per minute. So what happens? Basically, all the particles will get separated according to their weight. So then what will happen is that this infection will proceed and the viral coats were removed from the bacteria, all right, and they were agitated in the blender. The bacteria which were infected with viruses had a radioactive DNA and were radioactive. तो अभी जो भी बैक्टीरिया इन्फेक्ट हो गए बैक्टीरियोफेज यानी कि वायरस से, उनमें क्या था? उनमें था radioactive DNA, which indicated that DNA was basically the genetic material that passed from the virus to the bacteria. तो ये वायरस क्या करता है? वायरस बैक्टीरिया के अंदर जाता है एंड रेप्लिकेट कर रहा है. तो अगर अब डीएनए प्रेजेंट है उस वायरस बैक्टीरिया के अंदर, इसका मतलब है डीएनए इज द वन दैट इज has been passed from the virus to the bacteria. So the bacteria that were infected with virus that had radioactive proteins were not radioactive. तो अब जो बैक्टीरिया इन्फेक्ट हुए वायरस के साथ, all right, and that had the radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria. It was the DNA that entered the bacteria. क्योंकि जो प्रोटीन के radioactive थे वो जब it went inside the bacteria was not radioactive. DNA is therefore the genetic material that is passed from virus to the bacteria. So let us see some questions related to this. Hi brothers and sisters, how are you? So let us see some MCQs related to this topic. So what did we see? First we saw that how the transforming principle was set up. All right. So this is according to that transforming experiment was performed by. It was performed by Griffith. That is, uh, he basically what he did, he performed it with the Streptococcus pneumoniae. Pneumonia is caused because of, yes, the same question, Streptococcus pneumonia. So it infects all the mammals and the humans. All right. Genetic material is carbohydrate, DNA, fat, and lipids. So the genetic material that is present is the DNA. Now, coming to the properties of the genetic material. So from the ongoing discussion, now we have had a clear debate, all right, that it is the DNA that is the genetic material. And it was resolved by Hershey and Chase experiment. Now what happens that it became an established fact that DNA acts as a genetic material. So first Griffith just gave us the statement of transformation. Then what all Avery, MacLeod, and McCarthy performed was not readily accepted by the scientists, and they were questioned, and many their theories were rejected for many years. After that, Hershey and the Chase were the one who confirmed or verified the Griffith's prediction and the Avery, McLeod, McCarthy prediction as the DNA was the genetic material. All right. So, however, it in the coming years it became that some viruses RNA is the genetic material. So now the conflict is resolved. Now they have seen that DNA is the basic genetic material, but only in case of some viruses. RNA can be the genetic material. What are the examples? Examples of RNA as the genetic material is the tobacco mosaic virus, it is the bacteriophages, QB bacteriophage, and the HIV disease, which causes AIDS. All right. So this is an picture of the tobacco mosaic virus. This is of the bacteriophage, and this is the HIV virus. Fine. So a molecule that can act as a genetic material must fulfill the following criteria. तो जो भी एक मॉलिक्यूल लाइक डीएनए को हमने जेनेटिक मटेरियल कह दिया है, तो वो क्या करना चाहिए, ये फॉलोइंग क्राइटेरियाज को फुलफिल करना चाहिए. ऑलराइट, तो ये क्या क्राइटेरियाज है कि इट शुड बी एबल टू जनरेट इट्स रेप्लिका, दैट इज रेप्लिकेशन परफॉर्म होना चाहिए. यानी कि DNA has to undergo, they have to keep replicating so that the genetic material is produced and they are transferred from one generation to the another. It should be chemically and structurally stable. ठीक है, अगर ये स्टेबल ही ना हो तो ये पूरे बॉडी का जेनेटिक मटेरियल कैसे बनेगा. Then if it is not stable, what will happen? It will lead to severe mutations. So एक ही स्ट्रक्चर होना चाहिए डीएनए का, and it should not keep changing. It should provide scope for some slow changes or mutations. It should be structurally stable, but it should also be, there should be a scope for mutation, which is very much required for evolution. All right. It should be able to express itself in the form of Mendelian characters. What are Mendelian characters? Mendelian characters are basically all the traits and factors that will flow from one generation to another. So, because of the rule of the base pairing and the complementary, both the nucleic acid DNA and the RNA have to direct their duplications. That means DNA और RNA दोनों अपने आप replicate कर सकता है. So the other molecule in the living system, such as the proteins, fail to fulfill the first criteria itself. DNA and RNA replicate कर सकता है, but proteins जो है वो replication नहीं कर सकते अपने आप. Alright, and DNA and RNA replication क्यों कर सकता है? Because it has the rule of base pairing and it has the complementary structures. RNA is not predominantly RNA is not predominant and it is not very suitable as the genetic material because a very important question, that why RNA is not the genetic material. All right. So RNA is highly reactive and unstable. This is the reason of why RNA is not a suitable genetic material, because it is highly reactive and it is highly unstable. All right. Unstable and highly reactive. So the two OH groups, all right, two OH groups present in every nucleotide in RNA is basically a reactive group and it makes it labile and easily degradable. तो ये जो RNA में देखिए DNA and RNA में क्या डिफरेंस है? RNA is basically deoxynucleotide acid and जो DNA is deoxyribonucleic acid and the RNA is simply ribonucleic acid. ठीक है. तो अभी डीएनए में क्या होता है, इसके जो 2OH है ना, उसमें इज बेसिकली लैक्स ऑक्सीजन. ऑलराइट. तो इसमें इट विल लैक ऑक्सीजन, इसलिए वो डीऑक्सी कहा जाता है. बट आरएनए में जो 2OH ग्रुप है, इसके वजह से ये आरएनए को इजीली डिग्रेडेबल बना देता है. हाय भरत, हाउ आर यू? So RNA is also known to be a catalytic at its functions as an enzyme. तो RNA basically एक enzyme की तरह function करता है and therefore it is reactive and unstable. So the category that the genetic material should be able to should be very stable and it should not undergo many changes. So this criteria is not fulfilled by the RNA. And in RNA, uracil is present instead of thymine. All right. So uracil is basically less stable compound compared to thymine, and that is the methyl uracil of the DNA. तो uracil में जो RNA में एक और इशू ये है, इसमें प्रेजेंट होता है थाइमीन के जगह uracil, जिसके वजह से it is not very stable. All right. RNA being unstable mutates at a faster rate. तो RNA unstable है, इसीलिए वो जल्दी म्यूटेट होगा. See, for a genetic material there we required some mutation, but only in a very small amount that will affect the evolution. But RNA, since it is unstable, it will mutate at a very fast rate that is not suitable. Both के सेम स्ट्रक्चर है क्या? मैम नहीं, आरएनए और डीएनए के बहुत डिफरेंट स्ट्रक्चर है. कल का जो लेक्चर है वो होने वाला है स्ट्रक्चर ऑफ आरएनए एंड डीएनए पे. बट अब मैं आपको बता दूं, सबसे पहला तो ये है जो मैंने बताया था, 2OH ग्रुप में डिफरेंस होता है. डीएनए जो है वो डीऑक्सी होता है, यानी कि 2OH में ऑक्सीजन वो लैकिंग होता है, एंड आरएनए में वो ऑक्सीजन प्रेजेंट होता है. ऑलराइट. And the other difference is RNA has uracil and DNA does not have uracil. It has thymine. Alright. So consequently, virus having RNA genome and having shorter lifespan mutate and evolve faster. तो ये जो RNA genome जिन भी virus में है, they have a short lifespan and they mutate and evolve faster. This is one of the reasons that why we cannot find a proper medication for the HIV, because it has RNA as its strain. So the DNA is the predominant genetic material of the organisms. So what happens here? DNA जो है वो है predominant material of the organisms. Yes. So DNA is chemically less reactive and structurally more stable. Hence, it is the most suited. So, DNA can be clearly evident in Griffith's transforming principles. तो इसका जो स्टेबिलिटी है ये हमें पता चलता है ग्रिफिथ के ट्रांसफॉर्मेशन एक्सपेरिमेंट से, जब वो ट्रांसफॉर्म हो रहा है R स्ट्रेन से S स्ट्रेन में. ऑलराइट. इस प्रोसेस से हमें इसकी स्टेबिलिटी का पता चलता है. So just remember this line. It is many times asked in the MCQ of the NEET. All right. You need to remember this line thoroughly, that its stability is evident in the Griffith transforming principle. Now, heat which killed the bacteria at least did not destroy some of the properties of the genetic material. तो जब हमने उस heat killed S strain लिया ना, at least क्या हुआ कि ये जो genetic material है वो destroy नहीं हुआ. तभी जाके वो heat killed R strain से S strain में convert हो पाया. Alright, so what we see here that even by heating the DNA was not damaged, so it can be taken that the DNA is quite stable. This is how easily can be explained in the light of the DNA. That means this is how we can conclude that it is the DNA that is the genetic material. So the two strands being complementary if separated by heating. All right, अभी हम DNA के ये दो strand को separate कर रहे हैं, then even they will come together when the appropriate conditions are provided. Alright, तो what इसको हमने heat किया, ठीक है, then what happened, these two strands got separated. Alright, then again when the heat is reduced and stable conditions are provided, it will eventually come and form the strand again. Alright, it will eventually start coiling and again form the DNA. This was the Griffith's experiment. So what did we see here? Let me summarize it again. We had a non-virulent R strain that which resulted in a healthy mouse. The smooth virulent strain uh, made the caused the mice to die. Then we took the heat killed. हमने यही smooth strain जो pathogenic था उसे heat से kill किया. तब भी देखा कि माउस हेल्दी है, बट व्हेन द आर स्ट्रेन जिन दोनों में भी माइस हेल्दी थे ना, उन दोनों स्ट्रेन्स को हमने कंबाइन किया, तब हमने देखा कि द माइस डाइज. ऑलराइट, इसका मतलब ये ऐसे क्यों हुआ जबकि दोनों ही स्टेबल थे? बिकॉज फ्रॉम दिस ग्रिफिथ कंक्लूडेड दैट एंड इन द ब्लड सैंपल ना लाइव एस स्ट्रेन बैक्टीरिया वाज obtained. So इससे ग्रिफिथ ने कंफर्म किया कि there is something which transforms itself. Alright, so basically it was the DNA that is transforming itself. So the presence of thymine in DNA instead of uracil provides stability to the DNA. So see, this is how the structure of the RNA is and this is how the structure of the DNA is. So here this is basically the sugar of the thymine and this is that of the uracil. Alright. Now, hydrogen bonding between the purines and the pyrimidines. What happens? The hydrogen bonding and the stacking will make the DNA more stable. So basically DNA के अब ये जो A, G, C, T ये सारे बेसिस होते हैं डीएनए. इनके बीच में क्या होता है, जैसे G एंड C ले लिया, इनके बीच में देखने मिलेगा हाइड्रोजन बॉन्डिंग. ऑलराइट, सो दीज आर द हाइड्रोजन बॉन्डिंग एंड दे आर बेसिकली स्टैक्ड ऑन टॉप ऑफ वन अनदर. तो ये जो डीएनए है ये ऐसे होता है, ठीक है, तो बेसिकली दे आर स्टैक्ड अपॉन वन अनदर. Because of this stacking, they are more stable than the RNA. And DNA is capable of undergoing slow mutation and it also has a power to repair itself. So if the DNA is damaged, it will start repairing itself by itself. Alright? So that is what is needed to be a suitable genetic material. And both the DNA and the RNA involves genetic expression. Why? Because RNA can directly code for the synthesis of proteins and hence can easily express the characters. तो अब यहाँ पे क्या हो रहा है कि RNA जो है ना, यहाँ पे ये बोल रहे हैं कि RNA and DNA दोनों genetic expression है. Alright? Because RNA also will directly code for the synthesis of proteins. So here basically what is happening, DNA replicate करता है and ये transcription के बाद RNA form करता है which will lead to the synthesis of proteins. तो RNA जो है वो code करता है proteins के लिए and hence वो easily characters को express करता है. Hi, Adi. Hi. So DNA is dependent on RNA. Why? Because they synthesize proteins. And the protein synthesizing machinery evolves around the RNA. Alright? So the DNA being the more stable is preferred for the storage of genetic information, where RNA is better for the transmission of the genetic information. Alright? Let's see a few MCQs. So, based on which experiment DNA acted as a genetic material? It was based on the Hershey and the Chase experiment, where the bacteriophages were used. All right. Which base pair is present in the RNA instead of thymine? Instead of thymine, what is present in the RNA? It is the uracil. DNA is dependent on dash for the synthesis of proteins. DNA is basically for the storage of the genetic material, and for the or for the synthesis of proteins, what we require RNA. All right, because RNA acts as a enzyme. So this was the first lecture about how was the search of the genetic material. Now we have seen the history, that is basically how the genetic material was discovered. All right. So we have seen the discovery part. So where did it begin? It began with the experiments of Griffith. He stated the transforming principle. Avery, McLeod, and McCarthy, they treated them with enzymes, and they suspected that it is the DNA that was the transforming principle. Why? Because when they underwent their treatment with proteinase, RNAs and DNAs. The DNA ones, even in the DNAs one, we could find that there was no presence of the S strain, and hence DNA was said. But this again, it was not very widely accepted. Then came the experiments on the bacteriophage by Martha and Chase, and that concluded that the DNA was the genetic material. And in this lecture, we have also seen that why DNA is more stable, all right, more stable than the RNA. So these two things we have discussed in detail. We have seen all the points of why the DNA is more stable than the RNA. Thank you for watching this lecture. Please do like, comment your views and subscribe the channel. And also tomorrow we will be discussing about what is the structure of DNA, how is its composition, and how even RNA is, how the structure of the RNA resembles and differs with that of the DNA. Thank you so much for joining in, and do join me tomorrow at 11:30 a.m. See you all tomorrow.